Good morning. Welcome to today's MPG Primer session. I am very excited to introduce our speaker for today. Dr. Carl de Boer is an assistant professor in the School of Biomedical Engineering at the University of British Columbia. He did his PhD in the lab of Tim Hughes at the University of Toronto and was then a postdoctoral fellow here at the Broad Institute in the V's lab until the end of 2019, after which he moved to his current position at UBC, where he's starting his own very successful research group there. We're excited to have him speak today about the evolution, evolvability, and engineering of gene regulatory DNA. Carl has agreed to take your questions during the talk, so please go ahead and raise a hand or type in the Q&A box, and Diane will voice your question as they arise. So thank you so much again, Carl.
Thanks so much for the introduction, Sarah. So today I'm going to talk about this project. It was just accepted for publication, and it's been on the bioRxiv for the last while, so I'll show a link to that at the end where you can check it out for more details. So this work was started while I was a postdoc in Aviv Regev's group, and it was done in close collaboration with Ishit Devil Vaishnav, who's a very talented graduate student in the Regev lab, and he did most of the work that I'm going to talk about today. So as mutations change an organism's genotype, selection can act on the new phenotype, and this can change the frequencies of alleles in the population over time, with alleles that result in decreased fitness, like this green allele, dropping out of the population, and alleles that increase fitness, like the orange one, increasing in frequency. So today, we're going to discuss the relationship between genotype and gene expression level, which is kind of like an intermediate phenotype on which selection can act. So I love this image. This is from Wikipedia on the article on different types of selection. And it's, until you understand what's going on in these images, it's like one of the most confusing images I've ever seen for how to describe selection. But once you understand, it makes a lot of sense. So this is actually looking at types of selection acting on wolves from the perspective of the deer prey. So in this example, this is directional selection. So the primary selective pressure is whether or not the deer see the wolves coming. So in a woodsy environment, it's very dark, not a lot of light. These dark wolves can sneak up on the deer and eat the deer. So they are favored over white wolves, which the deer see coming. But in an environment with a lot of snow, the white wolves blend in with snow, and so the deer don't see them coming. So lower melanin is favored in the snowy environment and higher melanin in the woodsy environment. Now, in a mixed environment with both snow and woods, either dark or light coat colors are favored because the white ones can sneak up in the snow and the dark ones can sneak up in the woods, but this intermediate phenotype is disfavored. So this is called disruptive selection. In a rocky environment, stabilizing selection kicks in, where extremes of the phenotype, either too white or too dark, both of those are disfavored. Instead, the selection is trying to stabilize expression at this intermediate level. So selection doesn't have to be operating on the expression of melanin at all. So in an environment where the wolves are primarily hunting moles, then there's no selection on the melanin expression. The moles are practically blind. The wolves are already upon it by the time the moles have spotted the wolf. So the fitness is the same regardless of melanin expression. So selection on gene expression is happening in humans too, and genetic differences drive expression phenotypic differences, and these can sometimes result in disease. So GWASs and follow-up functional studies have really shown us that these SNPs in human enhancers and also promoter regions, these can actually drive different phenotypic diversity and sometimes result in disease. And the way these work is that it disrupts the enhancer in some way, we'll get into in a second, and that alters the expression of some gene. But because each SNP can actually alter the expression of multiple nearby genes, and this can happen in one or more cell types, and each cell type can alter the phenotype in multiple different ways. It's really complex to figure this out in human. There are a few awesome examples where people have really narrowed down what's going on here, but we don't understand it well enough to really get a global picture of how selection is acting on these elements. Studying evolution in humans is also quite challenging because of our long generation time and our desire not to be lab rats. So yeast is actually a great system to study the evolution of gene expression because so much is known about yeast and we have so much data. For instance, in this paper from Aran Segal's group, they measured the relationship between the expression level of the gene and the fitness of the organism for many genes. And we can see from these data, like in melanin and wolves, the relationship between genes expression and the organismal fitness can be pretty complex. And some of them, there's almost no relationship. Others, there's like a very small window where you're at peak fitness. And if you deviate from that, then it's really not good for the organism. Yeast is also great because there's very few cell types. There's essentially one or very few cell types, depending on who you talk to. And we also know where to look for gene regulatory changes. So today we're only going to discuss this one promoter region for yeast. And this is the -60 to -80 region. This is relative to the transcription start site. And this is really the major determinant of whether the promoter will be accessible or not, and a major location for transcription factor binding. So, but it's important to remember that there are actually many other mechanisms for controlling gene expression level, and we're not going to look at these at all. So really in the gene expression mixing board here, we're really just looking at one of many dials. Gene expression levels are encoded in part by the promoter DNA sequence, and mutations in the sequence can alter the gene expression level. As in humans, yeast use these proteins called transcription factors that read out the sequence of the DNA and modulate gene expression. For instance, this green triangle transcription factor prefers to bind this DNA sequence, whereas this orange triangle transcription factor prefers this other DNA sequence. All these factors bind the promoter together in pretty complex ways to regulate gene expression. These also interact with nucleosomes and chromatin. In yeast, there's over 200 of these transcription factors, and because of all these interactions, it's actually really complex. Nonetheless, I'm going to show you that we can approximate how all these factors work together using a neural network model, and we can do it reasonably well and use a model that captures this to make inferences about how these regulatory sequences evolve. So many evolutionary studies have looked at specific regulatory elements in detail, but our understanding of how regulatory sequences evolve in general remains incomplete. Many open questions remain. For instance, what does a typical regulatory mutation look like? How quickly can selection modify gene expression? And how did regulatory sequences evolve? We're going to answer these today, at least to the extent possible in our analysis. And we're going to make a couple of assumptions that are going to make our lives easier. We're going to assume that the only way that regulatory sequences influence fitness is through changes in gene expression. So the regulatory sequence isn't doing something else that's not regulatory. Next, we'll just recall that these expression fitness relationships, they depend on the gene. They're gene specific. And so learning the relationship between sequence and fitness directly is certainly much more challenging if we actually incorporate the gene, or it would necessitate incorporating the gene because it's so gene specific. Finally, we're going to assume that the relationship between sequence and expression is independent of the gene being expressed. That is, you could swap out one gene for another, keeping all the regulatory elements the same, and the expression level would be more or less the same. And experiments have shown that that's more or less true most of the time. So that's really all of the assumptions here. And since a sequence to fitness function would not generalize across these genes, we're going to focus on this gene agnostic sequence to expression function instead. So that's what we did. We learned the relationship between promoter sequence and gene expression. And then this was captured by a neural network. And we validated this model and showed that it correctly predicted gene expression levels. And finally, we use it to study regulatory sequence evolution. So I just want to take this chance to acknowledge that there are a whole field now of using machine learning to predict mutation effects, in particular with non-coding DNA, some of which I've highlighted here. This is now a growing field. As our models get better, this becomes more and more possible. And you should check out some of these papers if you're interested in learning more about the topic. So using the data that I'll describe in a second, we actually made two different models that both predict gene expression from promoter sequence in different ways. This one was described already in this Nature Biotechnology paper. It's an interpretable model. And basically the idea is that it's explicitly capturing the biochemistry for how transcription factors work. It's just going to come in a couple times when we want to know what's happening inside the black box, rather than looking in the black box directly, which is more challenging to do, but possible. We can just ask the interpretable model what is going on. And most of the time they have roughly the same answer. But the neural network that Ishit Daval-Vaishnav made is better at predicting expression. And so when all we care about is predicting expression, this is the model we want to use. And so we're going to use this for the vast majority of what I'm talking about today. In order to learn a function that predicts expression from sequence, we need a lot of examples of DNA sequences and their expression levels. So this is how we generated the needed data. We want to be able to predict expression from any sequence. So we start out by randomly sampling the space of possibilities. So here we clone in a random oligonucleotide into a core yeast promoter sequence that regulates this yellow fluorescent protein here. And this yielded about 100 million distinct promoter sequences, each of which is randomly sampled from the vast space of possibilities. We measured the expression level of these promoters by sorting cells by the fluorescence of the reporter gene into different expression bins. Here we use 18. We regrow the cells, and here we could see that it forms this nice ladder of expression levels after resorting the cells. So the highest has the highest expression, and so on until the lowest expression, showing that it's a reproducible expression program encoded by these random DNA sequences. Now we use targeted DNA sequencing to identify what promoter sequence was present in each of the 18 different expression bins. And this gives us this table at the end of millions of randomly generated DNA sequences and the expression levels they encode for. And this yields about 30 million sequences per experiment, which is on the order of a human genome's worth of DNA in every experiment. So here we have over 15 gigabases of new regulatory DNA that's measured using this system to date. So it really gives us a lot of power to create these expression from sequence models. So I'm not going to describe the neural network at all. It's in the preprint and will be published shortly. But basically, we designed it in order to capture how we think these transcription factors interpret DNA sequences. So the neural network.
May I interrupt you for just a second with a
Yes.
question that's been posted? So this is from Alex Blomendal, who says, nice to see you. And also says, sorry if you already said this, but I assume you're studying promoters only and would like to confirm that that's right. And as opposed to upstream regulatory elements, which are called, he believes, enhancers in the yeast context.
Yeah, so this is actually, this region here is kind of, it's overlapping the upstream activating sequences in yeast. It's from -160 to -80 relative to the transcription start site. So it's not the core promoter where the general transcription factors bind. It's upstream of that where most of the transcription factors are binding. Although sometimes there are more distal transcription factor binding sites as well. But those more distal elements are not present in every promoter. So we're focusing on something that is present in every promoter.
Great, thank you very much.
So eShit's model works really well. Here I'm showing the predicted expression on the x-axis versus the measured expression on the y-axis. And on the left are 10,000 random DNA sequences. On the right are 5,000 fragments. So these are 80 base pair chunks of actual yeast promoters that are measured in this system. And you can see in both cases from 92% for the native yeast promoters to 96% for random. The model explains that much. So as kind of a validation in the model, we used genetic algorithms to design sequences with either high or low expression level and using the model's predictions. And these sequences were on average more extreme than 99% of native promoter sequences for both high and low expression, and approximately 20% of both of them, they were more extreme than all native yeast promoter sequences. So it worked pretty well. We didn't really go through that many rounds of this genetic algorithm, so we probably could have optimized it more, but as a general proof of the ability to design DNA, this was good. So next we're gonna go more into the evolution side of things. So we're going to simulate some evolutionary processes. So first we're going to simulate neutral drift. So now there's no selection acting on gene expression. We're just going to introduce mutations and see how quickly expression changes. And the way we do this is we start for each sequence, introduce a bunch of mutations, but just select one randomly, and then do this for a total of n mutations. And then at the end of n mutations, we're going to compare the final expression level to the starting expression level. So on the y-axis here, this is the change in expression level, and the x-axis is the number of mutations we've introduced, and it's kind of a log scale. So here it goes up to 32 mutations, and remember, this is only an 80 base pair chunk of DNA. So you can see that the expression levels drift pretty rapidly, and at 32, you're almost as different as sequences that are just completely unrelated to each other. We validated this by synthesizing and measuring the expression of sequences with different numbers of mutations, 1, 2, and 3 mutations. These are the hardest to predict because they're the most similar to the original sequence, and we're predicting the change in expression. And you can see that the expression changes closely mirror the measured expression changes. The measured is actually slightly more dispersed than the predicted, and we think this is because of measurement error. More generally, mutations seem to follow a power law distribution where there are very few mutations that have a more substantial effect. But you can also see that across all these mutations, and here we're looking at all possible mutations to native yeast promoter sequences, even these other mutations in the middle, they can still affect expression in a decent way. And there's not really a clear cutoff here where you can say that these mutations affect expression and these don't. It's really a continuum that follows a power law. So why do so many mutations affect expression? So we'll look at the interpretable model, and because it can give some insight into what is going on here. This is an experiment we performed for the 2020 Nature Biotech paper. And what we did here is we took, for a random DNA sequence, we took a transcription factor binding site and slid it across. We actually did this for 6 different transcription factor binding sites, and for I think 3 different random sequence backgrounds, but I'm only going to show 1 today. So in this random sequence, we tiled this binding site at every possible location for both the top strand and the bottom strand. And for each of those sequences, we actually measured the expression level, which is shown here. And you can see that expression actually changes substantially as this motif is slid across both the top and the bottom strands. And because we have this interpretable model, we can ask, okay, how well does it predict this? It actually predicts these changes in expression remarkably well. So now we can ask this interpretable model, what's going on here? And because it's so it turns out it has almost nothing to do with this MGA1 motif that's being slid across the sequence. It's because as you are sliding this motif across, it's actually disrupting and creating transcription factor binding sites for numerous other factors. And these are affecting both expression directly and the accessibility of the DNA. So these functional transcription factor binding sites are remarkably common in random DNA and can greatly affect expression, even in completely random sequences. So maybe it's not so surprising that random mutations in regulatory DNA are frequently altering gene expression. So next, we're going to use this neural network to simulate strong directional selection. So how quickly can we exploit these mutations to evolve extremes of expression? So we simulated this strong directional selection by, again, you start with a sequence, generate all possible mutations, but this time you select the best mutation. That can be either the highest or the lowest expression level. We do them separately. Red is highest, blue is lowest. And then you get this trajectory of sequences where you have 1, 2, and so on mutations. And you can see that the predicted expression on the y-axis and number of mutations on the x-axis. As you increase the number of mutations, expression really saturates quite quickly towards these extremes of expression. And within like 3 to 4 mutations, you're pretty close to as far as you're going to get. And as before, we validated this, and now this is measured expression for these trajectories, and you can see that pretty much the same thing is shown with the measured expression as what the model predicted. You're saturating very rapidly at about 3 to 4 mutations, but you can evolve quite quickly from the starting point to get to the extremes of expression with very few mutations. So, but that represented evolution in only a single environment. And so we next asked how quickly expression could adapt to 2 competing objectives. So this is 2 different growth environments for yeast. We reran the same simulation, but this time we're trying to minimize or maximize the difference in expression between 2 growth media. So this is defined media here and complex media here, which are just 2 different yeast growth media. So here you can see the change in expression changes much more slowly. So here you can see when we're maximizing the expression change. I think I've swapped these labels here, but either way, it doesn't matter. It's just a flip of the y-axis. You can see the expression change just keeps increasing, and in either media, it hasn't reached saturation even after 10 mutations. So it's much harder to evolve very different expression levels in 2 different environments. So for the first conclusions, there's actually going to be 3. So for this part, we saw that many mutations alter expression, and expression changes follow a power law, with about 10% of mutations capturing about 50% of the expression changes. Most promoter sequences have some accessible large effect mutations. Very few mutations are required to go between the expression extremes in most cases. But satisfying competing expression objectives for multiple different growth conditions is much harder than just a single objective. And we speculate that this could be in part why the genome regulation in higher eukaryotes like ourselves is much more complex, because it's difficult to encode for all these different expression programs of all the cell types of our body in a single expression or a single regulatory sequence. So instead you have to encode it in multiple enhancers scattered throughout the locus. So next we're going to learn from natural variations. So we're going to look at this study, the 1011 Saccharomyces cerevisiae Genomes Project. And I love this study because they clearly must have seen the 1000 Genomes Project and they're like, well, that's human, so we have to do better. So they did 1011 cerevisiae isolates. So we used these genome sequences, and the first thing we did was identify all sets of orthologous promoter sequences. So these is all the natural variation present in all the cerevisiae isolates across the globe. So now we want to create a null model to represent kind of the variation we expect if there's no selection operating on these sequences. So we basically match the mutation number and Hamming distance, but this time scattering these mutations randomly throughout these regulatory sequences. And then for both the natural variation and the simulated variation, we predict the expression, distribution, and calculate just the standard deviation for each of these distributions. And then we come up with this expression conservation coefficient, which is just the log ratio of the standard deviation for the simulated and for the actual sequences. So if this is a high number, that means simulated had more variation in expression, which implies that the natural variation is constrained to have a specific expression level.
I have a question, or I wanted to voice a question that arose. I think I should have mentioned it a couple minutes ago when you were talking about sort of how this might extend to more complex organisms, but I will voice it now. And the question from an anonymous attendee is, since your neural networks are generated for one cell type, how might they perform in predicting expression for different cell types? And do you anticipate you'd need to build different neural networks if you were dealing with multiple cell types?
Yeah, well, so yeast, one of the reasons we're using yeast is because it essentially has one cell type, at least as we're treating it here. You can go into details about some weird life cycle stages, but they're not really weird. They're natural, at least life cycle stages, but we're ignoring those for now. We did test it in growth conditions, which are kind of like different cell types. So you can grow the yeast in different media and it has to change its regulation to match that media. So we did that, but still very few different growth conditions. But to answer the question more directly, yes, I think the rules are going to be, it's going to follow a similar grammar in different cell types. So thinking about humans, but the factors that are acting in those different cell types are going to be different. So probably you can share information between cell types. And actually some people have shown that this is a successful approach to learning cis-regulatory grammar in humans. We are still not there yet, but you do need to have some cell type specific information encoded in the model, whether it's one model or multiple models.
Thank you.
All right, so this is the distribution of the expression conservation coefficient for all yeast genes. And those to the right show evidence of expression conservation, and those to the left show more divergent expression. And we find that more than half of these genes show evidence of conserved expression, which is kind of what you expect. These genes with low ECCs correspond to traits that you might expect to be variable among yeast. So for instance, carboxylic acid and alcohol metabolism. So this could reflect that they're fermenting in different habitats. Whereas the ones on the right, these are much more core cellular processes like RNA and protein metabolism. And here you screw up expression of these genes and the yeast is dead. So you expect them to be highly conserved. So that's all consistent with what we know. So we validated this in a number of ways that I'm not going to get into. They're all in the preprint if you want to take a look. But for now, I'm just going to take this one interesting example, CDC36. So this is one particular promoter that has a low ECC value, and we looked at it in more detail. And what we found is that this is the expression level, and you can see it has this low ECC because it exists in the yeast population in these two different expression phenotypes, high expression and low expression. And this low expression actually evolved twice. You can see one lineage here evolved this low expression, and this other lineage here evolved low expression separately with two distinct mutations. These mutations are actually predicted to perturb the same transcription factor binding site. So this is a UPC2 binding site. It's a near perfect site in many of the strains, but in the one lineage, this critical C is mutated, and the other, this critical G is mutated. So Jennifer Molinet, a postdoc in Francisco Cubillos' group, restored the UPC2 binding site in one particular isolate that had this binding site perturbed. So she created the two mutations that brought it back, and she did it stepwise to create these intermediate steps. And we compared that to a strain, a sake strain that naturally had the binding site intact. And so here we're showing the predicted expression for these different sequences, and we measured the expression using qPCR and found that the correspondence was, honestly, if we did this experiment again, it probably wouldn't correspond as closely as it does now, just from natural variation in the measurement, but it really closely matches what was predicted. And we actually managed to find a phenotype for this too. So because of what we know about CDC36, we hypothesized that it might have something to do with transitioning between environments. So we looked at a transition between glucose media and galactose media. So the yeast has to adapt its transcriptional program to this new media. And we found that when you restore this binding site, it can adapt faster than with the wild type sequence for this wine strain. So that's pretty cool. So here we've kind of like reversed the problem of finding a function for these GWAS variants. We had the model, it predicted that these variants have a phenotype, and then we found the phenotype, as opposed to starting with the phenotype and then finding the genetics underlying it. So for part two, the expression conservation coefficient can be used to quantify selective constraint using our sequence to expression model and population genetics data. Selection on gene expression, I didn't show this part, but it can be shared over vast evolutionary distances, even to mammals. That's in the paper if you want to take a look. And we can find interesting examples of expression adaptation in yeast. So next we're going to talk about evolvability, which is the ability for an evolutionary system to generate new genotypes with new phenotypes. And it's really important in adaptation to new environments. So in this example here, we have these mutations, and they created these diverse genotypes with diverse phenotypes. So if we look at this system, this is a very evolvable system if everyone here has survived, because if selection starts acting on this phenotype, it's already got a bunch of diversity in the population and can easily generate more that will be fit in this new environment. If, however, selection has destroyed all these new genotypes and phenotypes, and the only one that can exist due to selection is this one genotype and phenotype, then we would say that is not an evolvable system. So we can use a neural network to quantify the evolvability of each sequence, and we quantify it as this evolvability vector. So this is how we do it. We calculate for each sequence of length L, all possible 3L mutations, and then use the model to calculate the change in expression for each of these possible mutations. So this is 3 times 80, so 240 different mutations. We sort this vector because the order is kind of arbitrary, so we just sort it to get this monotonically increasing change in expression function, and that is the evolvability vector. So now we're going to use an autoencoder to understand the landscape of these evolvability vectors. So we're using this architecture from this paper here from Smita Krishnaswamy's lab. So the way this works is you start with a database of actual evolvability vectors, and you run it through this autoencoder, and it tries to encode this in this small space, this small latent space, and then it decodes it out the other side from this small latent space to reconstruct the original evolvability vector. So the goal is to have the same thing output as was input, but pass through this low dimensionality archetype space. So we tested to see how many archetypes we need, and we found that 3 encoded most of the information present in these sequences, so we went with 3. The archetypes were pretty interesting. There are kind of 3 or 4 different archetypes, 3 that were encoded in the model and 1 that was indirectly encoded in this space. So one of them is this maxima expression level. So here it has a high expression level, and there are very few mutations that increase expression level. Most of them do nothing, but some of them can decrease expression level potentially substantially. At the minimum expression archetype, these have a low expression level. Very few mutations can lower it further, and there are a few mutations that can increase it. There's also this malleable archetype that's intermediate between the two, and here you have a lot of mutations that can increase it and decrease it. Now, right in the middle here is actually a 4th kind of robust archetype where expression level is pretty much fixed. There are very few mutations that can either increase it or decrease it. It's stuck in place. So we can show this again by plotting the expression and mutational robustness in this archetype space. So first expression, you can see there's high expression over here, and then there's a spectrum over to the other side where low expression is on the other side. So it's captured expression level inherently in this space, even though that was not encoded directly in the evolvability vectors. And this mutational robustness, this is just like the percent of mutations in the sequence that appreciably change its expression level. And you can see you can change expression level with many mutations near this malleable archetype and on the outside of this archetype space, but near the robust center, very few mutations alter expression level. And another thing that is encoded is that these malleable promoters, they can actually quickly navigate the archetype space, which is quite interesting. So this is just a measure of how much all these orthologous promoters from the 1011 yeast genomes project, how much they move around in this space. So for instance, here's an example where they're all over the space. So it has this high evolvability, dispersion, or if they're all in the same spot in the space, they have a low dispersion. So you can see in this robust center, they don't move around much at all. They're kind of stuck in the center, whereas on the outside, they move around much more. And then we'll look at two examples of this. So this is the DBP7 promoter. And this was the promoter that in S288C, the yeast reference strain, it was closest to the malleable archetype. And you can see across these different yeast isolates, their orthologs are all over the place for this promoter. For UTH1, however, this was the one that was closest to the robust center. Here, there's actually even more sequences across these different 1011 isolates. And they're actually more different from each other than for DBP7, but they're all right in the middle. They're all stuck in this robust center. So we can analyze these promoters using Ishet's model to see how these mutations affect the expression level of these sequences. And you can see for DBP7, within 2 mutations, you can go from the intermediate state that's present in S288C. You can increase the expression close to the max with 2 mutations and pretty much at the minimum within 2 mutations too. So it's a very dynamic promoter. UTH1, however, it's kind of stuck in the on position, and you require quite a few mutations to be able to actually like take apart the expression program encoded in the sequence. And actually the way that the model does this is also somewhat interesting. It actually creates repressor binding sites at the beginning. So you can see like over here, you could actually mutate these binding sites over here and restore a high expression level. So it's not even able to pick apart this high expression program. It does so by overriding it with repressor binding sites. So for the final conclusions, we can define evolutionary archetypes using these autoencoders. 3 archetypes explain the evolvability of most promoter sequences, and this archetype space can provide insights into the evolutionary history and future of promoters. And as I said, it's on the bioRxiv. It was just accepted into Nature, and so we're pretty excited about that. And Ishet, he was really a driving force for this project. So if you see him in the hallways of the Broad, he's still around there somewhere. I don't know if he's in the meeting today or not, but congratulate him on surviving the most rigorous peer review process I've ever been part of. I also want to really thank Moran Yasur, Lynn Fan, Jean Adekonis, Don Thompson, and Joshua Levin for some Ascomycete RNA-seq data I skipped over today. Jennifer Molinet and Francisco Cubios for the CDC 36 experiment I talked about. And of course, Aviv Regev, who is a fantastic mentor and was very supportive throughout this whole project, and also funding sources. And I just want to say before I take more questions that since leaving the Broad, I've started this research group at UBC, University of British Columbia. You can see we have some pretty fantastic views from on campus. And here I'm leading this group of fabulous researchers, and we're continuing this line of work, aiming to create similar data types, models, and analyses, but this time in humans, so that we can illuminate the genetics underpinning different human diseases. And hopefully I'll be able to come back and tell you about that sometime in the future. With that, I'd be happy to take any more questions. Thanks.
Thanks so much, Carl. That was a really amazing and thought-provoking talk. I'm going to, first of all, encourage audience members again to post any remaining questions in the Q&A. I will just ask the question of, I can imagine that in the wild, yeast populations are different in the environments that they reside in. And you've alluded to that a bit in terms of the modeling that in terms of a complex as compared to a defined medium. But I can also imagine natural environments differ in their predictability as compared to their fluctuations in terms of, I don't know, living in a particular agricultural crop as compared to maybe being in a more controlled environment in various ways. And I'm wondering if you have studied such populations or have predictions about what type of standing diversity might exist in one as compared to the other.
Yeah, it's actually really hard to study. I totally agree with you. Yeah, so we looked at basically one axis of natural, I don't want to say natural variation, but the changes in environment
Sure,
that
yes.
yeast are subject to, and that's just like the nutrients that it's growing on. And surprisingly, like in our experiments, we looked at the interpretable model for different growth environments, and not that much is changing.
Oh, interesting. Yeah.
So the expression program that's operating in like one media is almost the same as another media. So there's all these other things that can happen in like where yeast are naturally growing, like different toxins and such, and temperature fluctuations. So I don't really have a great idea for how those things are changing here, in part because the expression reporter system we're using, it relies on like steady state expression levels. It's a protein reporter. So if you were in a really dynamic environment, you would only see the average of that the reporter. So we're actually not able to study that at the moment, but it's definitely a very important and interesting aspect. And yeah, the way these transcription factors work that are very dynamic, for instance, like responding to changes in salt concentration, they happen very quickly. So they're like shuttled in and out of the nucleus and do all sorts of things very quickly. So I think that's actually going to change the answer quite a bit versus the equilibrium model that we're kind of creating here.
Well, I don't see any further questions. So I wanted to just thank you again for an amazing talk, and especially for joining us from several time zones away. That was really, really wonderful. And thank you for joining us so early in the morning. And we will look forward to hearing about the next stages of your work in the future.
All right. Thanks so much for having me. I can
you
go
so
back to bed now.
Exactly. Take care. Thank you, Carl.
All right. Thanks so much.
Thanks. Bye.